Lichen planopilaris, or LPP, involves inflammation and scarring of hair follicles due to a dysfunction of the PPAR-gamma receptor. Now, PPAR-gamma is responsible for lipid metabolism and how lipids are broken down. So a dysfunction of the PPAR-gamma receptor leads to a toxic lipid accumulation in the extracellular matrix. And again, extracellular matrix, to those who don't understand, that is just, think of it as the cellular environment of just fluids interacting with each other, where these cells are kind of distilled in. I want you guys to understand this fundamental point that in high enough levels, lipids become toxic in tissues in general, and it triggers an immune response that becomes very pro-inflammatory, and it just destroys tissues. And this is a phenomenon that's known as lipotoxicity. We can even see this in other conditions of the human body, such as non-alcoholic fatty liver disease, a condition where excessive lipid accumulation in the liver cells leads to inflammation, oxidative stress, and potential progression to liver fibrosis or cirrhosis, where the liver is just destroyed and you have a bunch of scar tissue. That thing can happen to hair follicles too if you have too much lipids that exist in that environment, and that those lipids can't be broken down. Again, the immune system comes in, it freaks out, and it destroys whatever is producing those lipids, but it leads to an overall destruction to the tissues itself due to a hyperinflammatory state. Lipotoxicity also contributes to type 2 diabetes, where lipid overload in muscle and pancreatic cells impairs insulin signaling and beta cell function. And finally, in atherosclerosis, lipid toxicity within arterial walls promotes inflammation, foam cell formation, and eventually that sort of plaque buildup, which increases cardiovascular risks, such as heart attacks and strokes. And these lipids can be triglycerides or, cholesterol in very high amounts. So these examples illustrate the systematic impact of toxic lipid accumulation on organ function throughout the body and disease progression. So when we reapply that sort of idea to the dysfunction of PPAR-gamma receptors in the hair follicle, right? When the sebocytes or the sebaceous glands lose their ability to break down lipids, what's going to happen? It's going to promote lipotoxicity. The immune system is going to come in and destroy that hair follicle. Now, targeting senescence and lipid accumulation may improve or maybe even prevent the inflammatory damages caused by your body, right? Your immune system, those lymphocytes. Now, I recorded the other part from another video, but I just want to jump in and do a more ad hoc video, so on the fly video, and I just want to kind of synthesize two ideas together, right? I want to link androgenetic alopecia, a non-scarring hair loss due to a sensitivity, rather, to DHT, right? I want to link that to, at least in some people, in some cases, possibly a confluence of factors that can lead to a scarring alopecia, primarily lichen planopilaris. And the two things that seem to link these two alopecias together, in my opinion, right, would be lipotoxicity. So what am I trying to express here? Well, this is a really good paper. It's a bit old. It's from 2004, and at the time of recording this video, it is 1:54 a.m., February 10th, 2025, and the Eagles have won the Super Bowl. If you're in the United States, you know what that means. But in any case, this paper titled, quote, Regulation of Human Sebaceous Glands, unquote, by Danae Tibutot, or Dr. Danae Tibutot, we can get an understanding of how important androgens are, right? When it comes to modulating, sebaceous glands. We get an understanding that DHEA seems to be important. This is, it can convert into various sex hormones. It can convert into androstenedione. But I think the more important aspect here is that although it can convert to androstenedione, obviously androstenedione can convert into testosterone. But I want to highlight a particular part in this article. I'm going to go to this section over here where it talks about dihydrotestosterone. I'm going to read this paragraph to you. It says, quote, Dihydrotestosterone is produced from testosterone within the peripheral tissue, such as the skin, by the action of 5-alpha reductase enzyme. Two isozymes of 5-alpha reductase have been identified. The type 1 isozyme is active within sebaceous gland. The type 2 isozyme is most active in the prostate gland, where it can be inhibited by drugs such as finasteride. Activity of 5-alpha reductase and 17-beta HSD exhibits regional differences depending upon the source of the sebaceous glands. In the skin that is prone to acne, such as facial skin, activity of the type 1 5-alpha reductase in sebaceous glands is greater than in sebaceous glands obtained from non-acne prone skin. This implies that more DHT is being produced in sebaceous glands from facial skin compared to other areas of the body that are not prone to develop acne. The net effect of the activity of these two enzymes is the greater production of potent androgens such as testosterone and dihydrotestosterone within sebaceous glands of facial areas that may, in part, account for the development of acne in these areas. Unquote. So what is going on here, right? This is an old article, by the way, but it seems to be that DHT has some sort of effect on sebaceous glands, or rather 5-alpha reductase activity has some sort of effect on sebaceous glands such that when you increase the presence of DHT, you can increase you can modulate sebaceous glands such that they produce more sebum. And it's in that production of more sebum that you can trigger acne. And what is acne but, this overproduction of oily, substance that leads to oily skin and eventually leads to an inflamed pore that has a bunch of sebum deposits inside of it, and a lot of white blood cells and pus, right? That's pretty much what acne is, just a rudimentary kind of explanation. So it's inflamed skin with sebum. And essentially, that's what seborrheic dermatitis is as well. Seborrheic dermatitis, you have that issue on your scalp. It's itchy and you have all this oil coming out of the scalp. And by the way, you have type 1 and type 2 activity, 5-alpha reductase activity in the scalp as well, right? And a lot of people will tell you that, oh, I have a DHT itch and I have such oily scalp. That's a huge complaint from many people that deal with androgenetic alopecia. So there are drugs that have been designed to treat this, right? We're going to look at this paper over here titled, quote, Efficacy and Safety of Topical Clascoterone Cream 1% for treatment in patients with facial acne, 2 phase 3 randomized clinical trials, unquote. And we can see here the mechanism of action of this steroidal anti-androgen called clascoterone. So what happens, right? This graphic kind of, gives us an idea of how acne occurs. You have excessive sebum production over here, right? And you have epithelial keratinization and inflammation, right? At the top of the follicle or the top of the pore over here. And over here you have a bunch of, bacterium, that are feeding off of this excessive amount of sebum that's being produced, and it's inflaming the skin. So if we go to segment B, we get an understanding that it is essentially referring to this unit of excessive sebum production as the sebaceous glands. And within the sebaceous glands, you see these little cells called sebocytes. Now, sebocytes have androgen receptors on them, meaning that they can respond to androgens. The androgen of concern here is DHT. Due to how potent it is, and because of how potent DHT is, it can possibly activate genes that are more involved in producing more sebum, or just, an abundance of sebum. So DHT binds to the androgen receptor, and it gets into the sebocyte's cytosol, which is the liquid portion of the cytoplasm of the cell. So this, if you're not looking at the screen, try to look at the screen, but I'll just try to describe it. You see this golden yellowish area? That's the cytoplasm, and the liquid inside of it is the cytosol. And that's where the androgen receptors are, right? So DHT gets in there and it binds to the androgen receptors of that cell, the sebocyte cell. And then it moves into the nucleus. And in your nucleus, it has your DNA, right? And it responds to androgen response sites. This DHT androgen receptor complex binds to those sites and it turns on certain genes. And these genes are responsible for producing sebum and cytokine production, right? And then it leads to inflammation, more sebum, and it causes a loop until something kind of cuts it out. Now, the mechanism of action of clascoterone is to compete with DHT in binding to the androgen receptor such that you reduce the instances in which DHT binds to the androgen receptor, and you're reducing the frequency in which that occurs. And if you do so, you're reducing the number of times you can message to the sebaceous glands to overproduce in sebum. So if you can reduce that frequency, you can sort of, ameliorate or try to treat the condition of, acne. And this is why, clascoterone, actually, it's currently in phase 3 clinical trial testing as Breezula for the treatment of androgenetic alopecia. Okay, so that's pretty interesting. But as you can see, this is why the drug got approved. It's pretty potent. It worked in reducing acne. Look at this. Patient 1, right cheek at baseline, and then patient 1, right cheek at week 12. Same thing for patient 2 and patient 3. It seems as if that clascoterone works at modulating the sebaceous glands. So this graphic helps us understand the importance of the androgen receptor, DHT, sebaceous glands, and sebum output, right? DHT stimulates the sebaceous glands to increase sebum that seems to be rich in cholesterol and triglycerides, which inflames the skin. And there's another aspect to this, right? I'll just show you this graphic real quick. It's essentially what I just said, right? I'll just show it here on the screen, but you can see clascoterone blocks the androgen receptor, and then it can block the signaling cascade that would ultimately lead to lipid synthesis and inflammatory cytokine production. But essentially, what I'm trying to show here is that lipids or lipotoxicity seems to be something that links androgenetic alopecia to scarring for hair loss, like lichen planopilaris and its variants like frontofibrosing alopecia, CCCA, so on and so forth. Because if you look at the histopathological features, right, they all more or less look the same, right, between LPP and FFA. And we're looking at this particular article over here, titled, quote, Frontiers in Lichen planopilaris and Frontofibrosing Alopecia Research, Pathobiology, Progress, and Translational Horizons, unquote. But we get the understanding that lichen planopilaris and frontofibrosing alopecia are kind of like the same condition, right? It says over here in this paragraph, LPP and FFA, two distinct branches of the same tree. And they talk about in this paragraph how they essentially, kind of have a similar histopathology, but they just, in a clinical setting, when the physician's observing the condition, the issue is occurring at a different, part of the body. So with frontofibrosing alopecia, it's occurring along the hairline, and with lichen planopilaris, it's, in the middle of the scalp or in certain parts of the scalp. So the top of the scalp, right? And conceivably, it could be in a more diffuse pattern because you have another condition called frontofibrosing, fibrosing alopecia in a distributed pattern. FADP, I believe that's what it's called. I'll pull it up over here. This is another sort of alopecia that exists. where you have a more diffuse kind of, issue at hand, right? I just showed you here on the screen. We're looking at this paper called Rare and Misdiagnosed Entity Fibrosing Alopecia in a Pattern Distribution. This is also the same thing, right? We're dealing with the same issue here. It says over here, the term fibrosing alopecia proposed by Du et al. Refers to a group of primary lymphocytic cicatricial alopecias, which are mainly distinguished by clinical pattern of hair loss, of which lichen planopilaris is the prototype. This group includes lichen planopilaris, frontofibrosing alopecia, FFA, FAPD, and other forms of sort of diffuse hair loss and scarring hair loss. So what does the PPAR-gamma receptor do? So in this paper titled, quote, PPAR-gamma agonist and their role in primary cicatricial alopecia, unquote. If we go to this particular graphic over here, we can get an understanding of a sort of mechanism of action, right? This is essentially describing an environment that occurs that leads to a destruction of the hair follicle. So what is this, right? You have genetic factors, or maybe viral factors, or something happens, maybe an environmental factor that causes for your PPAR-gamma receptor, right over here, to essentially be underexpressed. It's called a PPAR-gamma deficiency. And that causes for your sebaceous glands to become dysfunctional. So what happens? Well, inflammation and pro-inflammatory cytokines, they start to come about, right? And this signals to your body to release lymphocytes. The lymphocytes come in and they begin to destroy certain, parts of the hair follicle, particularly the sebaceous gland, as well as the bulge stem cell. And again, the stem cell bulge, or the bulge stem cell area of the hair follicle, controls whether or not your hair grows out of the follicle. So that's due to lipotoxicity. And that's a serious issue. And this graphic over here kind of helps us understand, there's this thing called PPAR ligands. And these are essentially, PPAR ligands are sort of proteins that function as transcriptional factors that regulate the expression of genes, right? So it could be some kind of a fat or whatever. It comes in. It could even be DHEA as well. DHEA is stimulatory to PPAR-gamma. It comes in. It binds to PPAR, right? It agonizes PPAR, and PPAR has to bind with another receptor called the retinoid receptor, and it forms a structure called a heterodimer. So it heterodimerizes, if I'm pronouncing it correctly. And these two, right, they bind together, and then they go to, again, they enter the nucleus of a particular cell. This cell is probably, let's say, a sebocyte cell, right? And over there, it begins to, activate certain genes at the PPAR receptor response site, right? Or PPAR response site, whatever. It activates certain genes that are responsible for metabolizing lipids. So by having a sort of agonist or something, a ligand that can trigger the PPAR-gamma, right? That makes it start to act properly, and then sort of kick it back into function, and it can possibly treat people that have lichen planopilaris, right? As you can see over here, I think in this particular paper, they're talking about specific drugs like pioglitazone hydrochloride that could be used to agonize or activate the PPAR-gamma receptor. So I'm skipping around a lot here, but yeah, essentially this is what we're dealing with here, right? Over here, DHEA, if it becomes underexpressed, it could cause PPAR-gamma to also become underexpressed too, because DHEA also stimulates PPAR-gamma receptors as well. So there's a lot of people that seem to think that, high amounts of DHEA or DHEAS will lead to, further hair loss, and that conditions like DUPA, DUPA is due to having extreme amounts of DHEA and DHEAS. No, it's not. DUPA could be caused by various things. You have to go get a scalp biopsy to see what's going on for you. For an individual, it could mainly just be a sensitivity to DHT. For someone else, it could be a confluence of factors. It could be, fibrosing alopecia in a distributed pattern. That could be a possible factor, right? It could be. It could be maybe just chronic telogen effluvium. You wouldn't know because DUPA isn't a condition that you get in a biopsy report. DUPA is just an aesthetic, right? But what I'm rambling about here is I'm trying to show that with excessive sebum production caused by DHT, some people who have issues with their PPAR-gamma receptor might, have an issue where their scalp becomes sort of lipotoxic, and that triggers lymphocytes, your white blood cells, the fighters, essentially, to come in and attack aspects of your hair follicle and inflame the area such that you have a confluence of factors. And this particular article over here talks about this as well, right? It talks about how people with frontal fibrosing alopecia, men or women, right, they may benefit from using 5-alpha reductase inhibitors merely because there seems to be an overlap between frontal fibrosing alopecia and female pattern hair loss and male androgenetic alopecia, or male and female pattern baldness. So I just want to kind of bring this to people's attention. If you're having an issue with an oily scalp, please go get that checked out. And I fundamentally think in some people, because of DHT, it can cause for that lipotoxic environment, and it can lead to further hair loss. So for me, this is just another reason why you would want to decrease DHT, so you don't have to deal with all that sort of issues, right? But I think I've rambled on long enough here. And yeah, I just want to say thank you guys for watching this video. Hopefully it's informative. Hopefully, it makes sense what I'm saying, and I hope to see you in the next one. Peace out. I'll just be including this section. It's from a video that I made a year ago, but I just want to include it because it's like a, a nice roundabout way of how all my research has kind of come together when it comes to looking at all this literature. So this is just going to talk about the squalene and the, the kind of lipid profile of sebum and triglycerides, cholesterol, and some other studies that kind of relate to androgenetic alopecia, along with seborrheic dermatitis. So, yep, hopefully you guys enjoy. See ya.
The dermatitis presents as scaly, sometimes oily, inflamed skin that can be itchy or even painful to touch. This is an inflammatory condition the cause of which is not well understood, although there does seem to be a genetic component, and Caucasian, particularly of Celtic descent, are most susceptible. It seems that the sebaceous glands attached to the hair follicles begin to produce a very rich form of sebum. The sebum contains fewer free fatty acids and squalene but increased triglycerides and cholesterol. In part, the trigger for may be androgen steroids. Times of hormone fluctuation, such as during puberty, can activate the onset of seborrheic dermatitis.
And there's actually an interesting study that talks about this entire process. The study titled, quote, "The Annual Changes of Clinical Manifestations of Androgenetic Alopecia Clinic in Korean Males and Females, an Outpatient-Based Study," end quote, by Woo Sung Jang et al., published in 2013, involved a total of 833 male patients, 385 female patients culminating in an overall count of 1,218 patients from both genders. Seborrheic dermatitis was identified as the most frequent associated disease in both male and female patients with androgenetic alopecia. Interestingly, the prevalence of seborrheic dermatitis in androgenetic alopecia was notably higher compared to the prevalence in the general population, which ranges from 1 to 3%. The connection between androgenetic alopecia and seborrheic dermatitis, while evident, has not been definitively established. The authors of this study make the point that it is known within the scientific literature that seborrheic dermatitis is related to the elevated production of DHT in affected areas. And they actually go on to reference an article from an academic paper titled Androgenetic Alopecia in Adolescents, a report of 43 cases, where it is even further established that there is some sort of connection and correlation between psoriatic dermatitis and androgenetic alopecia, as well as acne. So you have all these other conditions coming together because of DHT. This hormone DHT plays a crucial role in activating sebaceous glands, which is a significant factor in both seborrheic dermatitis and androgenetic alopecia, and also, forgot to add, acne.